Noir Plasma Chrome Bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.
Messenger ribonucleic acid is a single-stranded molecule of RNA that corresponds to the genetic sequence of a gene.
RNA is assembled as a chain of nucleotides. Cellular organisms use messenger RNA, mRNA, to convey genetic information.
Protein biosynthesis, or protein synthesis, is a core biological process occurring inside cells. Transfer ribonucleic acid is an adapter molecule composed of RNA, typically 76 to 90 nucleotides in length. In a cell, it provides the physical link.
Ribosomal RNA is transcribed from ribosomal DNA and then bound to ribosomal proteins to form small and large ribosome subunits.
Each nucleotide in RNA contains a ribose sugar, with carbons numbered 1 through. A base is attached to the 1th position.
Transfer ribonucleic acid, or tRNA, is an adapter molecule composed of RNA. It provides the physical link between the genetic code in messenger.
Small nuclear RNA is a class of small RNA molecules that are found within the splicing speckles and Cajal bodies of the cell nucleus.
Small interfering RNA, or siRNA, operates within the RNA interference pathway. It interferes with the expression of specific genes by degrading messenger RNA after.
As you can see, the sugar components are different. DNA uses deoxyribose, while RNA uses ribose. This difference affects how the base pairs bond.
An important structural component of RNA that distinguishes it from DNA is the presence of a hydroxyl group at the 2' position of the ribose sugar.
Nucleic acid tertiary structure is the three-dimensional shape of a nucleic acid polymer.
The naturally occurring enantiomer of RNA is dRNA. Like other structured biopolymers, one can define the topology of a folded RNA molecule based. There are more than 100 other naturally occurring modified nucleosides. The greatest structural diversity of modifications can be found in tRNA. Messenger RNA carries information from DNA to the ribosome. The coding sequence determines the amino acid sequence in the protein that is produced. The most prominent examples of non-coding RNAs are transfer RNA and ribosomal RNA, both of which are involved in the process of translation. Messenger RNA carries information about a protein sequence to the ribosomes. The mRNA is then exported from the nucleus to the cytoplasm. The earliest known regulators of gene expression were proteins known as repressors and activators, regulators with specific short binding sites. RNA interference is a biological process in which RNA molecules are involved in sequence-specific suppression of gene expression.
Long non-coding RNAs are a type of RNA, generally defined as transcripts of more than 200 nucleotides that are not translated into protein.
Enhancer RNAs represent a class of relatively long non-coding RNA molecules transcribed from the DNA sequence of enhancer regions. CRISPR is a family of DNA sequences found in the genomes of prokaryotic organisms, such as bacteria and archaea. The detailed study of each of these seemingly different processes elucidated that the identity of these phenomena were all actually RNAi.
Using single-strand RNA fragments, the COX functions as a key tool in gene regulation.
Next to be linked to regulation were exist and other long non-coding RNAs associated with X chromosome inactivation. Their roles to be silencing of blocks of chromatin.
The third major group of regulatory RNAs is called enhancer RNAs. It is not clear at present whether they are a unique category of RNAs.
In biochemistry, a metabolite is an intermediate or end product of metabolism. The term is usually used for small molecules. Synthesis of RNA typically occurs in the cell nucleus and is usually catalyzed by an enzyme, RNA polymerase, using DNA as a template, a process known as transcription. Introns are spliced out of pre-mRNA by spliceosomes, which contain several small nuclear RNAs. Or the introns can be ribozymes that are spliced by themselves. Primary transcript RNAs are often modified by enzymes after transcription. For example, a polytail and a 5' cap are added to eukaryotic pre-mRNA. Transcription is the process of duplicating a segment of DNA into RNA for the purpose of gene expression. Some segments of DNA are transcribed into RNA. In genetics, a promoter is a sequence of DNA to which proteins bind to initiate transcription of a single RNA transcript.
Helicases are a class of enzymes that are vital to all organisms. Their main function is to unpack an organism's genetic material.
An intron is any nucleotide sequence within a gene that is not expressed or operative in the final RNA product. Orthornivirae is a kingdom of viruses that have genomes made of ribonucleic acid, including genes which encode an RNA-dependent RNA polymerase. The innate immune system, or non-specific immune system, is one of the two main immunity strategies in vertebrates. An mRNA vaccine uses a copy of a molecule called messenger RNA to produce an immune response. The vaccine delivers molecules of antigen-encoding mRNA into cells. A primary transcript is the single-stranded ribonucleic acid product synthesized by transcription of DNA and processed to yield various mature RNA proteins.
The RNA-induced silent plex functions in gene segments.
In molecular genetics, a repressor is a DNA or RNA-binding protein that inhibits the expression of one or more genes by binding to the operator or associated silencers. A transcriptional activator is a protein that increases transcription of a gene or set of genes. Activators are considered to have positive control over gene expression. The tmRNA forms a ribonucleoprotein complex together with small protein B, elongation factor 2, and ribosomal protein S1.
Pwi-interacting RNA is the largest class of small, non-coding RNA molecules expressed in animal cells.
Ribozymes are RNA molecules that have the ability to catalyze specific biochemical reactions, including RNA splicing and gene expression. The hammerhead ribozyme is an RNA motif that catalyzes reversible cleavage and ligation reactions at a specific site within an RNA molecule. Nucleoproteins are complexes formed by proteins conjugated with nucleic acids. However, it could sometimes mean just a protein relevant to nucleic acid. Ribonuclease is a type of nuclease that catalyzes the degradation of RNA into smaller components.
This website makes learning so easy. I found all my resources here.